Hello friends, welcome to SV Union High School and Junior College, Pune. Standard 10, Science and Technology, Part 2. Lesson 1, Heredity and Evolution. Protein synthesis, transcription, translation, and translocation. All the living organisms like plants, animals, and man are made up of cells. A cell is a basic unit of all the living organisms. Every cell has a nucleus, which is a controlling center of the cell. The nucleus contains a supercoiled structure called chromatin reticulum. This chromatin gets converted into supercoiled and condensed structures called chromosomes during cell division. Each chromosome contains a strand of deoxyribonucleic acid or the DNA that is compactly packed on the nuclear proteins called as histones. Small segments of DNA are called as genes. These genes consist of the information and instructions to construct proteins. Let's see how these genes contribute towards the production of proteins. Protein synthesis, the process of production of proteins by the DNA through the RNA, is called as protein synthesis. Well, the central dogma of protein synthesis is that proteins are synthesized by DNA through the RNA. The genetic code on the gene present on the DNA is copied on the messenger RNA or the mRNA by the process of transcription. The mRNA later on translates this genetic code into a protein with the help of ribosomal RNA or rRNA and transfer RNA or tRNA by the process of translation. The process of mRNA synthesis from DNA is called as transcription. Transcription involves 3 stages. Number 1, initiation. Number 2, elongation. And number 3, termination. During initiation, when a structural gene to be transcribed or copied is turned on, an enzyme called RNA polymerase II gets attached to the starting point of the gene. With the help of promoter and the initiation factor, the RNA polymerase II enzyme starts the initiation process. The double helical DNA gets untwisted and both the strands are separated. Now, the RNA polymerase II enzyme continues to move along the DNA using the sequence of DNA, starts making a new strand of messenger RNA or the mRNA. It helps in arranging the ribonucleotides according to the sequence present on DNA and continues the process of elongation of mRNA up to the termination point. Termination of the formation of mRNA occurs when termination factors stop the formation of mRNA. So the mRNA is formed from the 5' end to the 3' end of the structural gene. An mRNA thus formed is a primary transcript and it has to be edited. That is, it has to be processed into a matured RNA. The primary transcript or the premature RNA consists of functional regions called as exons and non-functional regions called as introns. The introns are removed and the exons are glued together by ligase enzyme, and this process is called as splicing of mRNA. This processed mRNA is now ready to translate into a protein when it is protected by capping at 5' end and tailing at 3' end with a poly A tail. Thus, a matured, processed and single-stranded mRNA is synthesized by the process of transcription, but with two major differences between the DNA and the newly produced mRNA. Number 1, the arrangement of ribose sugar instead of deoxyribose sugar is present in DNA. And number 2, the arrangement of nitrogenous base uracil complementary to the nitrogenous base adenine of the DNA. Thus, the mRNA that is synthesized inside the nucleus comes out into cytoplasm through the nuclear pores of the nuclear membrane. As it comes out, it brings in the coded message from DNA. The message contains the codes for amino acids. The code for each amino acid consists of 3 nucleotides. It is called as a triplet codon. Triplet codon. The code for each amino acid consists of 3 nucleotides on the mRNA. This is called as a triplet codon. For example, AUG forms a triplet codon. UGA also forms a triplet codon. They represent the 3 nucleotides present on the mRNA now. Now, AUG is a triplet codon that codes for an amino acid methionine. Altogether, nearly 20 amino acids are coded by 64 codons, out of which 61 codons code for 20 amino acids and 3 codons do not code for any, and these are called as the stop codons. An amino acid consists of an amino group and a carboxylic group. They join together to form a long polypeptide chain when they are bonded with peptide bonds. These polypeptide chains get converted into proteins. The process of translation begins when the mRNA comes out of the nucleus into the cytoplasm. The protein factories called the ribosomes, made up of rRNA, comes and binds with the mRNA. The tRNA, which brings along the amino acid methionine with it, containing the anticodon UAC, binds with the start codon AUG on the mRNA. The ribosome contains 3 sites: acceptor site or A site, peptidyl site or P site, and exit site or E site. Next, tRNA brings in another amino acid and comes at the acceptor site. Here, the first amino acid is bound to the second amino acid by peptide bond. The first tRNA moves to the E site and leaves the ribosome. In this way, amino acids are supplied by the tRNA as per the message on mRNA. For this purpose, tRNA has anticodon having complement sequence to the codon on mRNA. This is called as translation. The amino acid thus brought in by the tRNA are bonded together by peptide bonds with the help of rRNA. During this process, the ribosome keeps on moving from one end of mRNA to other end by the distance of one triplet codon. This is called as translocation. In this way, the amino acid chain grows long and long to form a polypeptide chain. Later, the polypeptide chain undergoes changes to form what is called as protein. This is how protein synthesis takes place. When sudden changes occur in genes, it causes mutations. These mutations may either cause genetic disorders or may cause evolution.